In the Metabolomics core facility, we work with researchers here at NIEHS and actually across the entire NIH intramural research program to perform metabolomics analysis on biological samples. We're typically the first stop on their pathway to investigating a new problem. So they may come with a very simple idea or a very off-the-wall idea of, I have no idea what this gene does, so I'm going to knock it out, and could you tell me if there's a metabolic consequence?
Whatever biological questions they're trying to answer will help them look at metabolomic differences in those samples.
Oftentimes we go into an experiment not necessarily knowing what the outcome will be. And so our job is to help our collaborators really start to uncover what may be happening so that we can plan further follow-up experiments and try to get a better understanding of what may be happening in these disease models.
The Metabolomics core facility is an invaluable source of technology as well as expertise that has facilitated the highest impact of research conducted here at NIEHS.
We are very fortunate to have a lot of very high performance mass spectrometers that are the primary technique that we use. All of those are coupled to a liquid chromatography system, which allows us to do chemical separation prior to doing the mass spectrometry analysis. And in addition to that, we also have an instrument capable of doing ionability. And with all three of those techniques at hand, we have really an immense amount of capability to both measure what is there in a sample, as well as dive down and figure out how much of something is there.
They actually identified a glutamine pathway in our system that I hadn't identified before. It would have been a lot more challenging to find this glutamine metabolism dysfunction in our glycerol knockout polycystic kidney disease model.
And they've helped deploy both targeted and non-targeted approaches, which can be difficult, I think, in other places where the instrumentation might not be easily accessible. So it's been really awesome to have that here.
So often we don't necessarily know what we're looking for going in. Our goal isn't necessarily to prove a point, it's to uncover the full scope of what's there in the sample. That's important because so often we don't even know where we need to start in order to make progress, maybe treating a disease or limiting an exposure that's causing a disease.
We can work with investigators to design their experiments for getting good metabolomics data and results that they are looking for. We're really part of the research that they're doing and not just sample handlers.
In the future, we can do more of a targeted assay, which is specific pathways that we want to look at instead of getting this huge broad overview, but we can also potentially be more accurate with the amounts of each metabolite in the samples.
NIEHS is probably at the forefront, certainly metabolomics. I think we've started to establish ourselves as a leader in that area. I think if there's a need for a mass spectrometry and metabolomics experiment here at NIEHS, we are very well equipped to handle most of those requests.
Metabolomics is really kind of a new frontier now. So having them here to add on to these other components of my project like epigenetics and making these new connections, I think truly we are really at the edge.
The field is progressing very quickly on all fronts. It's a hardware, software, how to handle, process the samples, and abstract the most information you can out of the sample itself.
I am so excited to get to the lab, turn on the lights, check on the instruments, and really start assessing what do we need to do today.
The enthusiasm is off the charts, they're really, really nice people. They have an open door policy, which is very helpful to come in and discuss questions in person. There's a lot happening in the Metabolomics core facility, so they're always working on something new and cool.
Always available, willing to teach, and willing to help you at every juncture to make sure that you know what's going on with your data, what's going on with your samples, and what you can interpret from the readout of the instrument.
I am personally really passionate about human health and the environment, looking at how exposure to those contaminants can impact our biological processes and human health. Those are always something that I'm personally interested in.
You have everything here in one facility, And I think that that is invaluable. You can talk to so many different experts here. It really can enhance your knowledge so that you can perform better science.
I'm interested in applying the skills, the knowledge that I've learned using these instruments to better understand what makes us tick and what also makes us sick, and if we can improve the human condition.